Thank you.
Thank you, Mr. Chairman. Thank you to the organization to have given me this lecture just 24 hours ago. So, Professor Cittadini, for you, I would have done even 2 hours before. And this is because Professor Cittadini is really an example of science, of medical dedication, and it has been for us in the last 40 years. I had the chance to meet him like 30 years ago, 25 years ago, and he invited me to my first oral presentation here in Palermo, I think in 94, 95. So really, it's a pleasure. It's a pleasure to be here. I have a very difficult topic because, by the way, I'm not really a pioneer in embryology. I'm too young for that. It's just that, so I always bought media already prepared. So when I received a phone call from Professor Cittadini, oh, I have to make a lecture, I say, okay, no problem. I have a lot of slides in my computer. Oh, it is about the challenge of developing media. Oh my God, developing media? I don't know. So I have changed a little bit the approach of the presentation because I'm not an expert in developing media, but I'm an expert in developing ideas. So I hope that you are going to follow my ideas in this presentation. So the title is Mimic the Nature. So this is a slide representing what is happening inside the reproductive tract. And it's really incredible what we learned in the last 2 days, how much science and dedication was needed to understand all the process, from ovarian stimulation, ovulation, timing of ovulation, and then how fertilization, the morphology of the embryo, the changing the morphology of the embryo, and finally to reach the blastocyst stage and implantation. Incredible, an incredible science with an incredible discovery. And around this, there is also the environment of the embryo. It's not enough to know about the morphology of the embryo, but if we want to reproduce what is happening in vivo, we have also to know what is needed from the embryo. So by looking back to the natural conception, and even if this slide is in 2D and not in 3D, unfortunately, we can feel how much the dynamicity of this process. There is an embryo that is changing its morphology, is making a lot of meiosis and mitosis, and is becoming a full cell, a cell, a morula, and then a blastocyst, and this is done moving all the time through the reproductive tract. Dynamicity is for sure part of the story. Mimic the nature. Are we doing any dynamic culture today, 2018? Absolutely not. So what is missing for sure is the dynamicity. Because what we do, I think everywhere in the world, is to put the embryo in a drop, surrounded by oil, covered by oil. And this is completely different from what happens in vivo. So first of all, the volumes are very different. In vivo, you have just a small film of fluid around the oocyte and then the developing embryo. While in vitro, we have this big drop, can be 20 microliters up to 80 microliters, and the embryo is just put there for days and days. In vivo, we have a maternal tissue that is thinking about everything, providing effectors and nutrients, removing waste, providing oxygen and CO2, taking care about the moving embryo. And probably in the reproductive tract, the condition, the environment of the embryo is changing also all the time, while in our culture dish, everything is very static. So it's incredible, because since the first culture of an oocyte in 1956 in a mouse model, many achievement has been obtained in science. We went to the moon. We were able to crack the DNA, to make the full sequential of the DNA. But still, to perform an embryo culture, it's a demanding task. And we have not made so much discover about what is needed for the embryo. How can we fully exploit the quality of the embryo that we are culturing in vitro? So since 1978, when the first idea to put an embryo on a dish was made to today, we have generated probably much more questions than answers. Should we culture the embryo in group or individually? Should we need co-culture? Which is the right oxygen concentration? Nobody knows. A supplementation of the media, sequential or single, as the chairman has suggested before. Renewing, should we renew the media during culture or we should keep it? I can tell you that we don't have answer to all those questions. And I will try to understand why. So first of all, we are in a different situation. This is more or less the composition of a culture media. Just copy a slide from one of my friends. And what we know for sure is that we are in a super physiological level. We are in vitro. And all this component has been decided according to the composition of the fluid inside the reproductive tract. But here we have some problems, because when the media were first designed, they were related to some factors and phenomena that has not been really confirmed in pregnant mammalians and not in pregnant women for sure. Some constituents that are present in the oviductal fluid may be detrimental. It's not the same to be in vivo or in vitro. Some factors that are necessary in vitro may not be necessary in vitro. In vivo analogs may have adverse effect in vitro. And we tend to prevent any kind of stress, but probably mechanical stimulation that occurs in vivo and other kind of control stress may even be beneficial. So the biggest problem is that animal models not always really mirror what happens in human. So, are we sure to know what is used in our lab? This is the question I made to myself. Do I really know what is in the culture media that I'm buying and using every day? First of all, every single media is different from the other. And it's quite incredible, but there are differences even in the most simplest element that are provided by the companies. Leader scientists would disagree on the right osmolarity, pH, oxygen tension, which is needed around the embryo. And what is even more terrible when thinking about improving and understanding is that more than 80% of the major producers of culture media do not provide a list of components and the exact composition of their media. So we are using something without really knowing what is inside. And this is really the opposite of what André van Strategen told us yesterday. Remember that André says that already in 1993, he was inviting a lot of people to see ICSI in his center. He opened the door, and I'm one of these persons that was so lucky to be in Brussels in 1994. By the way, I met Filippo Baldi at that time, and it's 22 years that we are working together. So opening the door, explaining new technology, disclosing the composition, this is the basic of improvement, understanding, and research. And this is not what we have today, because we don't know the real composition of the media that is provided. But we know which are the theories behind the media composition. And we have basically two different theories. The single-step media, so the same media for the whole culture, it's called the let the embryo choose. So it is believed that we put in the media everything that is needed at tolerable ranges, and the embryo itself will adapt to the environment. But we have a second theory, which is called back to nature hypothesis, and this is why sequential media has been produced, because the idea is to mimic the changing needs of a developing embryo in the media, but should approximately be at the same concentration of what the embryo is naturally exposed. So this is the science behind the two types of media that are actually in the market. And I also report a third theory that is important when thinking about our procedure, which is the quiet embryo hypothesis. And again, it's about stress. Are we sure that we should avoid stress? It's not for sure that a very active metabolic embryo the metabolism of the embryo is very active, is good. It's not sure because probably the best embryo has a quiet metabolism. So do we have evidence saying that single-step media or sequential step media is better? We don't have any evidence. I just pick up one of our meta-analyses that we did like 2 years ago, 1 year ago, and you see there is no clear evidence that it is improving. We can improve the embryo development and the embryo implantation by using one of the two hypotheses. The embryo is very plastic and probably behave more or less in the same way when we look to clinical pregnancy. This is live birth. When we look to blastocyst formation, we may find some differences. The single drop. Is it the best way to go? It's 40 years that we are culturing the embryo in a plastic dish and a single drop. There has been some attempts to try to change this. So to culture in tubes instead of dishes, to introduce microfluidics, new system like the GO system, the well of the well. But only with the introduction of time-lapse, we really change our way to culture the embryo. And what time-lapse has introduced, with or without the combination of the well, is the undisturbed culture system, because you don't have to take in and out the embryo to look to its morphology. So this is, to my knowledge, the best improvement that we have done in the culture environment in our labs, and this is applied in many centers today. Do we have really evidence that this is useful? I don't know. We have a low-quality evidence. The first meta-analysis showing in randomized control trial, but it seems that the live birth is improved, but we cannot discriminate if this is due to the culture itself or to the selection procedure. So we still have to work on this. But this is a piece of the puzzle. If we want to avoid to disturb the embryo, this technology is for sure logically improving our culture system. I don't know if I really need a randomized controlled trial, because this is logic. It's like a parachute. We don't need a randomized controlled trial if to jump with a parachute or not from an airplane. So undisturbed culture system is surely a big improvement in our setting, in the everyday life of a lab, not only in the setting of a randomized perfect control study, but in the everyday life is certainly an improvement. But my presentation will be mainly focused on that, because the only question that I receive every time I talk about culture system is, are we sure that we don't have long-term effect on the embryos and on the babies by the exposure of the embryo to the in vitro condition? And the question is important, because we are culturing the embryo in a very specific period. And the early embryogenesis is not only morphological changing of the embryo, but look how much we have epigenetic issue related of that particular period. Methylation and demethylation occurs during gametogenesis and early embryogenesis. And if the embryo has to adapt to an inappropriate environment, this may cause changes in the epigenetics, in the transcription, in the metabolism, in the cell allocation at the blastocyst stage, and this may have long-term effect on the blastocyst. And why are we discussing about this? Because we have a lot of examples in animal models that in experimental condition, if the embryo are exposed to particular environment, we may have increased birth weight, abortion, neonatal death, postnatal malformation, and this is true in sheep, in cattle, and in mouse. And what is the mechanism involved of this? It is probably related to epigenetics. So in the animal models, we know that specific condition may affect the life of the offspring. So this is very important to know. But what about human? Probably humans are very plastic. The plasticity of a human embryo is different, because in fact we have a lot of data supporting that there are no long-term consequences of in vitro culture. And although there has been some reports that some pathologies are increased in IVF, none of these were really attributed to the in vitro culture. And this is because we have some bias. First of all, the infertility itself, that is a risk factor. So when we compare two different populations, it's very difficult to understand what is the reason of what we see. But for sure, human embryo adapt very well to our labs. Is it a pure chance? Are human beings belonging to a lucky minority? Or we cannot rely on that for sure. We have to study. We have to understand if and when we can affect our embryos. So I pick up just a couple of studies to show you how difficult it is to understand if culture media and culture environment may affect the babies. So the randomized control trial, this is the only randomized control trial, very well designed, 2016, the group of Claire Kurst. I hope no Netherlands people are in the room because I'm not able to pronounce that. This is the group of Masterbrook. It's okay? That's not too bad? Okay, thanks. One is there. Okay, very nice study. The primary outcome measure is live birth rate. So the study has been powered for live birth rate, and more than 800 couples were involved. Randomization was done, and media 1 was better than media 2. We had a significant difference in clinical pregnancy rate and in live birth rate, although it didn't reach significance. But if you see not only the difference in pregnancy rate, but also the number of embryos that could be frozen in media 1 as compared to media 2, you can see that we have more embryos even in media 1, which means that more probability to have a cumulative live birth rate. So it seems that media 1 was performing better according to our outcome measure. Media 2, it's quite different from the other media because there are some different components that were absent. Okay, now the authors also look to the neonatal outcomes. Here I have to make a little lesson of statistics. It is very important because secondary outcome measures in randomized control trial are often very misleading. And I will try to understand why, to explain you why. So you have a study population that is randomized. So at this point, first step, the group are similar because they are the results of a randomization. So the primary outcome measure is coming directly after the randomization. So this and this could be compared. But if you have a difference in live birth rate, this group is different from this group, 184 pregnancies against 159. So it's like, to make not a scientific way to say it, but these are the survivors. This group that obtained a live birth, although they were in a condition that was less efficient for embryo development, for embryo cryopreservation, they survived and were able to have a pregnancy. It is a selected population of patients. So we have a first bias, uneven distribution of live birth. And then also, we have a difference in singleton and twins. So when the authors were comparing singleton versus singleton here, 3 steps lower than the randomization, these two groups cannot be compared anymore. It's 167 babies compared to 136. We are highly underpowered for neonatal outcomes. Of course, it was not the primary outcome of the measure. And we have 2 bias of selection. And it is not because of the authors, but it is quite impossible to design a randomized controlled trial to look to neonatal outcomes because you cannot randomize pregnancy. You cannot retrospectively randomize pregnancy according to the culture media on which the embryo were exposed. So randomized controlled trial, when dealing with neonatal outcome, are highly underpowered and subjected to bias. Okay, so what can we do? We have to rely on big cohort studies. This is the only way to go. On very large cohort study, probably we have to rely on national registry or international registry. In Italy, there is a very nice project to have a single cycle record of a national registry. This will be able to do what? Not only to collect data, but also to adjust for confounders. So if you have a huge number of data and you have all the confounders, you can adjust for confounders and try to make some understanding of what is happening. And yesterday there was a question, is ICSI more risky for baby as compared to IVF? Okay, ICSI, not because of, for me, not because of the manipulation itself, but ICSI is potentially more risky than IVF because you have extra manipulation of the oocyte, and oocyte is a very sensitive moment. You have to denude the oocyte, you have to expose the oocyte to the inverted micromanipulator for minutes during the injection itself. So when we look to this incredibly huge study performed on the English data, 144,000 of cycles, all adjusted for confounders. And confounders is not only maternal age, but it's also duration of infertility, cause of infertility, previous pregnancy. We have at least 11 confounders that we have to put to understand in which population we are. It was quite incredible to see that ICSI had a positive odds ratio for live birth, probably because we have more embryos and we can better select the embryo, but also for neonatal outcome. In the multivariate analysis, the odds ratio for both preterm birth and low birth wave in singleton IVF were reduced when women, rather than donor, eggs was used and when ICSI was used. The opposite of what we were thinking, but ICSI was an extra manipulation that could affect the birth weight of the babies. So how to explain this? It's very difficult, probably because if you have more embryos in a population at risk because of a male factor, you can better choose the embryo, and probably this is why it's behaving better. We don't know, but it's very reassuring when using ICSI instead of standard IVF. And this is the biggest study, cohort study that is published. So how can we explain now that human embryos are very tolerant to in vitro conditions? Because millions of babies are born, and we're still discussing if there is any effect on neonatal outcomes. So it means that there is a great plasticity and a great tolerance of human embryo in different conditions because all our labs are different. None of us is making the same culture condition in our lab. So the tolerance is because humans are very different from animals. And just to make an example, you know that in some animals, you need an exact match between endometrium and embryo. So in bovine, not only you can only transfer blastocysts, but also in the right part of the ovary, next to where the ovulation was obtained. And this is completely different in human. In human, you can transfer even a 2p stage embryo and obtain a pregnancy. It means a lot of plasticity, a lot of tolerance. And I can make many other examples of that. So we are for sure in a lucky situation. But it is our responsibility to continue to develop research and ideas. Because this is a generous gift of nature, but must be a limit. I mean, this tolerance has for sure a limit. And if we may cross this limit at any time, the consequence will be realized years later. Because we don't have a study that we can design properly to understand the effect of anything that we do on the babies, except if we make a huge study and we see retrospectively the data. So this is a great responsibility, and this is what the pioneers have left to us, developing ideas on embryo culture research and keeping in mind our responsibility. And we should start thinking about the future. And in fact, we have some new possibility. Dynamicity is possible in a lab by introducing microfluidic device. Maybe difficult for embryology, maybe demanding task, maybe expensive at the beginning, also time-lapse was. But for sure, we have to try to think to better mimic the nature, to increase, to fully exploit the potentiality of the embryo that we have in our lab. And in parallel, we should also continue to understand the needs of the embryo. And probably this can be done by making different measurements in the culture media, which is a source of different things, including DNA. And this is what I see for the future. So thank you very much for your attention. I have to thank two persons. One is here, the other one is in Rome. The two persons, Danilo Cimadomo and Gemma Fabozzi, that spent the last 24 hours preparing this slide. It's not me. Danilo is here. Danilo was able to follow the meeting and prepare the slide. You see, young people are able to do two things together. We should rely on them for the future. Thank you very much.